welcome Gary Moore.
Thank you for the introduction and the opportunity to share with you today some of my thoughts and research efforts in the area of artificial photosynthesis and solar fuels. So before the introduction, I had this quote on the screen, and it points to a future where we can change our energy infrastructure from one that's dependent on fossil fuels and oil to one where we move into clean technologies based off the process of photosynthesis, where we're able to capture the energy of the sun. So this rather visionary article came out in Science in September, but it wasn't September of this year. This was September 1912. So these concepts of mimicking photosynthesis have been around for quite some time, as have been research areas and efforts in this field. So although I think that you'll see some of the things that people are working on nowadays are very different from some of the concepts in 1912 of how to do photochemical energy conversion. Does anybody know when oil was first discovered? There it is, 347 AD in China. It wasn't until 1815 that oil was first produced in the United States, and at this time it was a byproduct of the chemical industry. Places like Dow Chemicals were literally digging for salt, brine, to build up the chemical industry, and there was this black stuff they had to deal with and get rid of, oil. In 1859, there was the first drilling of a successful oil well in the United States in Titusville, and this was very related to a series of pioneering experiments that took place at Yale University by Benjamin Silman. He was a professor in the chemistry department, and he looked at the various heats and enthalpies of combustion for these fractional distillations from this black oil. This is a picture taken from the Drake well in Titusville, Pennsylvania, not too far from where I was born, but a long time before I was born in the temporal sense, anyhow. This is actually the second Drake well. The first one was destroyed in a fire. And this is 1859, and this is the world today. We live on a planet with a population past 7 billion. We have an energy demand for the planet of 16 terawatts, and that's projected to double by 2050. And there's a serious question of how we'll meet this energy demand in a sustainable fashion. I think this picture is nice, and it illustrates, in a sense, global energy consumption, and also that it's not spread throughout the globe equally. Right? You can see that our planet is very much more lit in the northern hemisphere than the southern hemisphere. But there is hope in meeting these future energy demands, and that hope comes in the power of the sun. The sun is an abundant energy source with 120,000 terawatts is the rate striking the surface of the Earth. However, it is a diffuse energy source. So some time ago, about a year ago, Gary Brudvig and I wrote a review article where we started to look at some of the best research cell efficiencies available. So these are solar to electric. And one of the things I think comes out of this type of graph is you can see, for the most part, these various approaches to solar to electric conversion have been improving in efficiency over time. But bear in mind, this is only efficiency and doesn't take into account cost. So if you were taking into account cost in this graph, I think what you'd find is that all of these essentially would come together into a single trajectory within 20% of each other. But notice we can have very cheap cells that operate with low efficiencies, or very efficient cells up past 40%, right, that operate, but these come with a significant price cost. This is another illustration of a calculated air requirement at 10% efficiency for solar to electric to meet the entire U.S. electrical demands. So this is a 100-mile by 100-mile grid. Now that's a large area, but bear in mind that human efforts have built structures on these scales, especially when you think about highways and roads. That's less than a quarter of this area. But the problem is not one of only capture and conversion. When you think about scaling these technologies up, a real problem becomes storage. In other words, the sunlight, like most renewable energy sources, is not constantly available. The sun sets, and you need a way of storing that energy. And in this sense, photosynthesis offers inspiration for large-scale deployment in that regards. It's mastered the chemistry of converting sunlight into chemical fuels. However, this process is not capable of powering human demands for 7 billion people. This is an iconic image of the Keeling curve, and what it shows is a function of CO2 concentration versus time. And what we can see is this oscillation on an annual cycle. And this is because of photosynthesis. This is net CO2 uptake and output from the process of photosynthesis. And it's swamped by human activity. This is CO2 correlated with the burning of fossil fuels. And this is known, right, from radio-labeling isotopes experiments. This is a slide adapted from an MRS bulletin report and some input from Tom Moore. They came out of some discussions that we've had. Looking at the big picture, again, I mentioned this 120,000 terawatts striking the Earth. Now, photosynthesis transduces solar energy at 90 terawatts. Again, that's a big excess of human energy demands, but there are various processes, downregulation processes, which at the end make this process rather inefficient. And so when we go through things like internal conversion, regulatory dissipation, CO2 assimilation, at the end we're left with about 4 terawatts for primary food consumption. And this number is still in big excess of the number you would get if you multiplied 7 billion times a 2,000 calorie per day diet. In a sense, artificial systems or human engineered systems can already outcompete photosynthesis in terms of efficiency. And so this is a slide that illustrates the efficiency of 3 photovoltaics wired together and coupled to a commercial electrolyzer versus the natural process of photosynthesis. So electrolyzers operate at pretty high efficiencies, upwards of 70% commercially available today. And photovoltaics you can buy commercially that operate between 10 and 20%, so we'll say 15. That's overall 11% energy conversion efficiency. By comparison, the process of natural photosynthesis, again, will vary somewhat depending on the organism, but in most cases, less than 1% efficient. In other words, the sun, solar light, convert it to a burnable fuel. There's also questions you can think about stability, the warranty. You can currently get about 30-year warranty on photovoltaics. I'm not sure how to think about the warranty of something like a leaf, somewhere between 30 minutes and 3.2 billion years. If you're interested in these sort of analogies between artificial systems and human systems, I point you in a direction of a recent article in Science by Blankenship and co-authors that really look into the intricacies of making some of these comparisons between natural systems and human-engineered systems for energy conversion. So although human-engineered systems can be quite efficient, they do come with a cost. And they typically use elements in the catalyst that are not abundant, things like platinum and iridium, where nature has figured out the trick of using more abundant first-row elements like cobalt, nickel. You can invoke diagonal relationship manganese, which is at the heart of the oxygen-evolving complex in photosystem 2. Now, there have been attempts in human-engineered systems to make low-cost devices for splitting water and producing a fuel in the form of hydrogen. This comes out of early work in 1977 by Honda and co-authors, where they used the direct illumination of TiO2, titanium dioxide, with UV light. And when this cell is completed with a platinum counter electrode, you can produce hydrogen at the other side. Now, the quantum efficiency for this device, in other words, the number of photons to give rise to charged carriers, is 10%. But the overall efficiency, the power conversion efficiency, in other words, the amount of sunlight that is captured in the form of a fuel like hydrogen, is less than 1%. And this is really in large part due to the poor spectral coverage of TiO2. TiO2 is a white material. Everything that you see that's white usually has TiO2 in it for the most part. It's very abundant, very cheap material, but not good at maximizing coverage of the available photon flux coming from the sun. And that's illustrated in this graph, where I'm putting 3.2 electron volts, which is the band gap for this material. And you can see it just covers a small fraction of the available energy coming from the sun. Now, in 1991, Gretzel and co-workers reported a device that builds off of TiO2, but it's a dye-sensitized solar cell. It uses a molecular dye that absorbs light, and the HOMO and LUMO of this dye are such that it brackets the conduction band of the TiO2 material. So now, this device can absorb visible light, electrons can be transferred into the semiconductor material, and again, at the counter electrode where the cell is completed, a redox couple is used. In most cases, something like triiodide iodide which allow for a cyclic electron transport and the conversion of solar energy to electricity. So this device operates with an incident photon conversion efficiency, or quantum efficiency, near unity. In other words, all of the photons that strike that die give rise to charge carriers for the most part. And in terms of energy conversion efficiency, it's pretty good. 11% is not bad. The downside is in that in using these dyes to bracket the conduction band, this dye is no longer thermodynamically capable of driving things like water oxidation. It's capable of oxidizing low potential species like the triiodide-iodide redox couple and can produce solar to electric, but it does not address solar to fuel issues. In 2009, Youngblood and co-workers from Malouk's group at Penn State reported a system that in many ways is a hybrid between the dye-sensitized solar cell and the Honda cell, which I showed earlier. In this case, the cell again uses a molecular ruthenium dye, but it's connected to a water oxidation catalyst, iridium oxide nanoparticles on 2 nanometer size scale. So in this system, photoexcitation leads to a initial charge separated state where it's completed again by a platinum counter electrode where hydrogen can be produced and oxygen formed at the anode side. Now, one of the nice things about this work is Malouk's team did a really good job, I think, of going through and studying these compounds and their kinetics using pump probe spectroscopy. So they're able to look at the individual rate constants for many of these electron and hole transfer processes, as illustrated in this energy level diagram shown to the right. One of the things that should immediately stand out about this is the rate constants for the forward process of reducing the photooxidized pigment with the iridium catalyst. This is 2.2 milliseconds, whereas recombination, in other words, electrons from the TiO2 quenching the dye and the process running in essentially the backwards fashion, has a time constant of 370 microseconds. So this essentially runs backwards, almost an order of magnitude faster than you would want it to run in the forwards direction. That's a bad thing for these types of devices. However, it does capture many key elements of a device that you would like for thinking about doing water splitting. Overall, the system operates with an external quantum efficiency of 4.5 times 10 to the minus 3 and a quantum efficiency of less than 1%, and can produce photocurrents on the order between 10 and 30 microamps, depending on the applied bias voltage. So in most of these experiments, you require still a small bias voltage in order to produce hydrogen at the counter electrode. So it's not completely photo-driven. It's a photo-assisted reaction. So in this way, it's plausible that nature may offer insight on how to take what are essentially cyclic electron transport systems and think about building linear electron transport systems for fuel production. These are energy diagrams associated with bacterial type 2 reaction center, which is a cyclic electron transfer process. In other words, the last electron transfer step in this chain is to reduce the photooxidized pigment. Nature had to evolve systems like this to form systems which could handle water oxidation, as is in the case of photosystem 2, which does carry out water oxidation and produce essentially the thermodynamic equivalent of hydrogen. This can be things like NADPH, ATP, high-energy molecules that are used as energy carriers, which capture that energy that was in the sun. One thing that's clear, you can see, was this necessary to move the photopigment to more oxidizing potentials that are thermodynamically capable of handling things like water oxidation, or these species that are kinetically and thermodynamically difficult to handle. And so this does raise the question, can we use strategies like this for converting solar to electric systems to solar to fuel systems by taking analogy from nature? In 2012, I reported a series of compounds that are high potential dyes for applications in solar conversion in dye-sensitized solar cells. These consist of porphyrin rings, which are analogous in structure somewhat to the tetrapyrrolic pigments of chlorophyll, which are the natural pigments in photosynthesis. However, these dyes, the artificial dyes, bear these bis pentafluorophenyl groups, which are electron withdrawing and make the dyes highly oxidizing. In other words, these electron withdrawing groups destabilize the oxidation of the species, or the formation of a cation on the species, so that if you are able to oxidize these species, what's left behind are very powerful oxidants, thermodynamically capable of driving water oxidation. So based on the optical and electronic properties of these dyes, which I showed in the previous slide, we can start to draw energy level diagrams where we look at the ground state potential and estimate the excited state potential based on the color of the dye, or where these dyes absorb light. We've made a range of these complexes, and we can compare these with the traditional ruthenium-type dyes, which are used in a dye-sensitized solar cell. And again, you'll see that for the most part, these dyes which we make are much more oxidizing than the ruthenium-3, ruthenium-2 couple in a traditional dye-sensitized solar cell dye. These energy level diagrams are typically drawn with more positive values at the bottom and more negative values at the top, as I have illustrated in this slide. Now, together with Rebecca Milo and Charlie Schmuttenmeyer at Yale University, we looked at a number of photoanode materials, TiO2, that have been sensitized with the dyes illustrated in this slide, and studied them by terahertz spectroscopy. This is a pump probe technique where we can excite the dye with visible light and then probe with terahertz. Now, the thing about terahertz is terahertz radiation can be absorbed by mobile electrons in the conduction band of various metal oxide materials. So the greater the terahertz absorbance, the higher the population of mobile electrons in a given semiconductor. So it gives you a direct handle at looking at how efficient a given dye will inject into a given metal oxide material. So these are results illustrated here on TiO2 photoanode materials. So the one with the greatest delta terahertz in black corresponds we're using a typical ruthenium dye. No surprise, these are very efficient at ejecting electrons into TiO2 as they are designed. For our high oxidation potential porphyrin dyes, the only one that really gives rise to a significant delta terahertz is the green zinc derivative of these high potential porphyrins. And this is expected from our energy correlation level diagrams, where it's the only one other than the ruthenium species that really has an excited state potential poised more negative of the TiO2 conduction band. But we can switch that metal oxide material to something like tin oxide. And tin oxide would have it's shifted much more positive. Tin oxide at plus 0.05 versus TiO2 at about 0.57. And these now illustrate using, again, the ruthenium dye, just to show you that we know how to make dye-sensitized solar cells. These are IV curves looking at the current voltage response using something like triiodide iodide. These give rise to cells that, in our hands, operate with an energy conversion efficiency of about 9%, close to the values reported by Gretzel and co-workers. But if you were to switch that redox mediator to one that's more oxidizing, something like tribromide-bromide redox couple at 1.05 volts, the cell completely shuts down. And that's because these types of dyes are thermodynamically incapable of oxidizing these highly oxidizing redox couples. And the same is true on tin oxide for the most part. If we were to replace that dye now, though, with a dye with a potential thermodynamically capable of handling bromide, we can see large increases in the performance of these cells when using the tribromide-bromide couple, shown as a dashed line, instead of the triiodide-iodide couple. So what we've done is re-engineered these cells, which were designed to operate using relatively low potential mediators, that now perform better using high potential mediators. The idea being that this is a proxy for oxidizing something that's difficult to oxidize, like water. These cells, however, do not have a catalyst. And catalysis will be important when you're thinking about especially things like water oxidation, where you have OO bond formation is a difficult reaction, and you need to bring together two electron-rich oxygen species. Furthermore, the first step in this process, right, would be energetically inaccessible, and many of the intermediates would be bad for the cell. So there are a number of examples of human-engineered catalysts in the literature. For a long time, there was not that many. The first example of a molecular catalyst for water oxidation is a ruthenium blue dimer. This was later followed by a manganese dimer that was reported in 1999. Since that time period, in these last few years, there's really been a plethora, a huge breakthrough. The doors were kicked down in the development of molecular catalysts, both for water oxidation and reduction reactions, and significant progress has been made. I don't have time during this talk to go into various breakthroughs in that field, but needless to say, one of the fastest catalysts around at the time when we were doing some of this work with modifying dye-sensitized solar cells was one reported by Bob Crabtree and co-workers that's an iridium CP star complex. Now, this catalyst has a highly electron-donating species that's necessary for stabilizing high-valent iridium. And likewise, the carbon-iridium bond is a very strong bond, so it's a robust material. It operates with a turnover frequency of about 1 per second. So that's how fast it will put out an oxygen molecule as a function of time. So this catalyst was initially developed for use in solution in the presence of chemical oxidants like cerium. It's not clear always that catalysts designed in this fashion will work in electrochemical experiments, but in 2011 we showed that we could make an analog of this catalyst that bears a carboxylic acid for functionalization on TiO2, and that indeed the catalyst remains chemically active, electrochemically active when immobilized on a surface. This is key for incorporation into photoelectrochemical devices. Further, we've shown that we can incorporate or co-sensitize TiO2 with both our high potential dyes and the use of these iridium CP star complexes mobilized on a surface. Now, this material, optically, the material that's co-sensitized with both materials, shown in red, looks more like the dye sensitized material. In other words, the catalyst itself absorbs very weakly in the visible region, and most of the light-absorbing properties come from the dye. Now, from an electrochemical standpoint, looking at CV measurements of this compound, the trace in black shows a voltammogramic response for TiO2 that is deposited with the iridium catalyst. And what we see is at potentials positive of 1 volt, the onset of large current densities that we can correlate with the formation of oxygen. Whereas with the co-sensitized material, we see, again, an onset of a current density, but at lower values. Now, some attenuation of this current is expected, as on the TiO2 surface, sites that were available for the catalyst to be deposited are now occupied by the dye, which is carrying out light absorption. And you can see that the slide shown in green, or the trace shown in green, is for a TiO2 that's sensitized with only the dye. And in that case, there's no onset of current associated with catalysis or oxygen production. Likewise, in photochemical experiments, it's only when all the parts are together, the dye, the catalyst, the TiO2, that with the illumination of light, we can see the onset of relatively large currents on the order of 30 microamps. So at the time, this was a rather unique approach to thinking about building water oxidation cells based on dye-sensitized solar cells, in that it uses a co-deposited method. In other words, the porphyrin and the iridium are not covalently attached to each other in previous work I had showed you with the ruthenium dye and an iridium oxide nanoparticle. This really opened up avenues for looking at exploring the stoichiometric ratio of catalyst to dye in these systems. Now, another interesting observation is that none of these systems, in comparison with photosystem 2, does the light-absorbing species directly interact with the catalyst. In photosystem 2, there's a redox mediator, a tyrosine species, which potential is finely tuned so that it's in between the potential for water oxidation and the potential of the oxidized dye. Now, tyrosine potential is thought to be finely tuned in these complexes by its close proximity to a histidine residue, His 190, in which it forms a hydrogen bond that tunes in the properties of this material. Our group and other groups have reported analogs of these tyrosine-histidine pairs. This species shown in blue is one where we have a phenol species bound to imidazole in such a fashion that it can form a hydrogen bond with the imidazole. This has significant effects on the electrochemical properties of these materials. If you take, for example, a phenol that is not attached in such a way that it can form a hydrogen bond with the imidazole, right, the potential for these are out near 1.4 volts and are irreversible. In other words, when these species are oxidized, they form a species which is highly acidic, pKa on the order of -2. And so oxidation is coupled to deprotonation. This phenol will deprotonate to the bulk, and the materials is no longer available to reduce on the counter sweep. Now, for these species shown in blue, it's a different situation. We've now trapped the proton at the site of electrochemical activity. Oxidation of the species results in the structure shown on the bottom, where the proton shuttles and makes an imidazolium cation and doesn't release it to the bulk. So by trapping that proton at the site of chemical interest, the species is now completely chemically reversible, and the potential is tuned to one that's appropriate for acting like a mediator between the light-absorbing species and a potential water oxidation catalyst. We've gone on to make systems like this where we've attached that high potential mediator to highly oxidizing dyes and look at their electrochemical properties. Importantly, the electrochemical properties of the phenol are not perturbed upon coordination or attachment to the molecular dye. And we study these complexes in photochemical studies via EPR. So upon photoillumination, we can see that electrons are injected into the TiO2 material, with electrons being localized both in the bulk and in the surface, and that we can form an organic radical. By going to higher resolution D-band EPR, we can get a direct idea of or observation of the molecular species that are involved in that organic radical. A large portion of the holes are indeed localized on the phenol, and this is resolved in the anisotropy of that species, where we can pull out the GY, GX, and GZ tensors, and these are values which all match closely those found in natural photosynthetic complex and tyrosine systems. Furthermore, these systems show a unique temperature dependence. At 80 Kelvin liquid nitrogen temperatures, approximately 80% of the holes are localized on the phenol complex. In other words, secondary electron-proton coupled electron transfer is efficient, while at low temperatures, 4.2 Kelvin liquid helium, the holes are more equally distributed between the phenol and the porphyrin radical cation species. And when we allow the species to warm, we go to a situation where, again, 95% of the holes are localized on the phenol and will remain there when cycling the temperature back. These trapping of a high-energy state we think is due to restricted nuclear motion in these complexes, and is very reminiscent of an observation in natural photosynthetic systems in tyrosine D, which has given rise to much speculation in regards to the mechanisms of how these biological systems operate. The question being, is this high-energy species one that results from a proton-coupled electron transfer pathway or a pure electron transfer pathway? So in the proton-coupled electron transfer process, both the electron and the proton are transferred together, giving rise to an intermediate species, as illustrated here, where we have a neutral phenol bound to a midazoleum cationic species. Or a situation where we have electron transfer from an initially deprotonated phenol that gives rise to this species that then can relax upon warming due to histidine motion or relaxation of the tyrosine and histidine. To think about that in an energetic perspective, this stepwise versus concerted, I don't want to get too bogged down in the nomenclature of this field. But one way of thinking about this concerted processes are that they involve a single transition state. In other words, the electron and proton transfer without forming an intermediate. Now, temporally, what's the situation there? It's a little bit more vague. But clearly, in proton transfer, electron transfer, or electron transfer, proton transfer, we're forming high-energy intermediate species. So this offers mechanisms, again, from biology of thinking how to transfer electrons with low energy requirements. We'd also looked at these species not only in the protonated form, but in a deprotonated form. By titrating these complexes with a base, we can selectively deprotonate and form the complex as shown here. We have a pretty good handle on not only the connectivity of these compounds, but also the three-dimensional orientation of these compounds based on nuclear Overhauser spectroscopy. So we can see if we deprotonate the phenol, there's a twisting of the dihedral angle to form the phenoxide that's now bound to an NH versus an OH bound to an N-lone pair. So what's unique about these structures? Well, what's unique about these structures is it offers another direct handle at a molecular level of controlling the redox potential of these species by having an ionizable proton present. So again, the species in blue is for the neutral form, where the phenol was hydrogen bound to the N-lone pair of a benzimidazole. For the anionic species, we have a huge shift in the redox properties. And we can also look at the protonated species, where we disrupt the hydrogen bond by protonating the N-lone pair and shift the electrochemistry of this species in the more positive direction. Likewise, we've also looked at building complexes of this type where the system is covalently attached again to a photochemically active chlorophyll-like pigment. Here now, instead of using our highly oxidizing species, these are decorated with a mesotyl group. And the reason we did this is that we wanted to create a system where upon photoillumination, the electron transfer does not take place. So in other words, looking at the optical and electronic properties of this particular porphyrin, the relay for the excited state, in other words, when you shine light on this dye, the ability for it to accept an electron and make the porphyrin anion radical in this case is at plus 0.6 volts versus SCE. This is thermodynamically then incapable of being reduced by the attached phenol species. In other words, if you analyze the energetics of the systems, this is uphill by 340 millivolts. So the species should not undergo photoinduced electron transfer. But what we've shown is that by ionizing the species, right, we now have another way of controlling the photochemical process. We can go from a relay at 0.94 volts to a relay at 0.26 volts, which is indeed, should be thermodynamically capable of reducing the porphyrin by the same value but opposite sign, minus 340 millivolts. So it's an illustration of the control of these systems at a molecular level. Now we can look at, again, the photochemical properties of these systems upon illumination for both the protonated complex and the deprotonated complex. And as one might expect from the energetic analysis, there's a quenching of the porphyrin fluorescence when the species is deprotonated, which is consistent with electron transfer from the deprotonated species. Now, we can do that, but the thing about it is this species that's formed in the deprotonated complex is thermodynamically incapable of water oxidation. So this observation, which was inspired initially design features from nature, also gives us insight into how biological systems may operate. So again, these complexes, which are human-designed and molecular systems, closely resemble those found in the natural system of photosynthesis, where you have a phenol, again, bound to an imidazole, or a phenolate bound to the NH of an imidazole, as the A2 and A1 reactants in a proton-coupled electron transfer process or a stepwise process. It's interesting then to compare these results to other photochemical systems. An earlier example of such a system is one reported by Hammerstrom and co-workers in 2000, where, again, using a ruthenium dye and a complex that's covalently attached to the ethyl ester of tyrosine. Photoillumination of this complex in the presence of an electron scepter, like methyl viologent, can induce a proton-coupled electron transfer process where the ruthenium cation is reduced by the attached tyrosine. There's a very beautiful study where they've looked at the rate of electron transfer in these systems as a function of pH. And what they find is two regimes. One regime at lower pH, and a second regime that's much faster at higher pH. It's interesting also to compare these results in human-engineered systems with ones observed in biological systems. So here is looking now at oxidation of tyrosine in photosystem 2. Again, where we see a very similar relationship. Two regimes, one at low pH and one at high pH, where the electron transfer becomes much faster and is not dependent on pH, and one at lower, where it's slower but has a pH dependence. The interesting thing to point out is the breaking point on these two. One, the breaking point occurs at about a pH of 10, and another one, the breaking point at about pH of 7. So we think these reflects at some level the pKa's of the species associated with this. In other words, as you increase the pH, you will deprotonate the tyrosine species, and the electron transfer is no longer coupled to proton transfer, and thus is not a pH-dependent process and is much faster given the change in the potential for the tyrosinate versus tyrosine species. Whereas in the natural photosystem, this break occurs about pH 7 and may reflect protonation of the imidazole to imidazolium rather than the tyrosine and tyrosinate species. So this is a system where it gives us insight to biological systems, but what is the potential role in human-engineered systems for addressing energy transduction? Here's an example of a system that was reported in 2012, again from the group of Maluk and co-workers, where the ruthenium dye, which was used in the initial system, is no longer directly attached to an iridium oxide nanoparticle. In this case, the iridium dye is now separate and is attached to TiO2 through an organic linker, but the iridium oxide particles are capped with an analog of these high potential mediators, these tyrosine histidine complexes. And in these studies, incorporating the tyrosine histidine as an electron mediator has a significant effect on increasing the current density of these systems for water oxidation, a fourfold almost increase in the current density and associated efficiencies of these complexes. So one of the problems with all of the devices that I've showed you so far is, in a sense, they're single junction devices. As you move the potential of the light-absorbing species to more positive values in order to accommodate things like water oxidation, you lose the power of the reducing species to either inject into things like TiO2 or handle things like the reduction of a substrate to form a fuel. And this is one area where biology offers significant insight on how to handle this, and that's not having a single junction system, but a tandem system where you have more than one light absorber. So in natural photosynthesis, we have PS2 and PS1. So again, similar situation. The photosystem 2, the P680, the light-absorbing chlorophyll-like pigment, is thermodynamically poised to oxidize water, but the excited state relay is not thermodynamically capable of reducing NADPH or even things like protons to hydrogen. What it does is rely on a second photosystem that those electrons are dumped into and pumped up a second time, right, to then drive chemistry on the reducing side to convert the solar energy to fuel. So although photosynthesis is successful at using this tandem systems, one of the things it did not do was take advantage of maximizing the coverage to the solar spectrum by offsetting the optical properties of the second photosystem. This has been illustrated, again, I point to those who are interested in these sort of concepts, to a paper by Blankenship and co-authors in Science last year. So how do we identify promising photocathode materials? In other words, in order to capture and convert solar energy, one of the problems of doing this is that carriers moving through a potential in a device, they must have a threshold. And so where should we put these thresholds? This has been worked out by initially Shockley and Quasar for a single junction system. The optimal sweet spot would be at 925 millivolts. And this is really just a trade-off of integrating across the AM 1.5 solar spectrum and asking yourself, do I want a few photons at high energy, or do I want all the photons at low energy, essentially? Right? So it's a trade-off between the number and the energy of the photon. There's also been analysis of this for multi-junction systems or tandem systems. Hanna and Nozick and others have looked at this. So for a tandem device, you can increase the efficiency with the theoretical maximum of 46%. But in that case, you'd like to stagger the threshold of the light-absorbing species. You'd like one that absorbs light at approximately 760 nanometers, and another one that absorbs light at 1305 nanometers. So this notion of using multiple junctions in order to gain in the spectral coverage, and even moving to things like semiconductor materials, is not a new one. I share with you here an example from Turner and co-workers in 1998, where, again, they report a multi-junction cell that operates with quite high efficiency for photochemical water oxidation, 12.4%. More recently, 2011, Steve Reese and Dan Nocera and co-workers have reported a system based on a multi-junction silicon cell, which oxidizes water and produces hydrogen. This cell operates at a much lower overall energy conversion system, 2.5% compared to 12.4%, but it captures many of the key functional elements of a leaf, namely the use of earth-abundant materials, such as cobalt and nickel in this case, for building these types of devices. Now, in scaling these sort of to commercial application, there are other issues which will naturally come up, but this is a nice piece of work in that regards. So I shared with you today a number of examples of approaches to thinking about doing photochemical storage by being inspired from the processes of photosynthesis. But all these are examples of isolated components. No one has really built a device that's efficient, that's low cost, and is robust. It captures all of those elements. You can usually capture two, but no one's captured all three in a single material that does the complete process. This is a quote from Melvin Calvin. It's time to build a natural artificial photosynthetic system to learn what works and what does not work, and thereby set the stage for making it to work better. With the little bit of time I have left, I'll share with you some of the work that I've been involved with more recently at JCAP. The mission of JCAP is to demonstrate a manufacturable, scalable solar fuels generator that operates with efficiencies 10 times greater than current crops. It does so on the premises that a group of researchers working together can accomplish more faster than investigators working on their own in individual projects, and that's not to understate the value of individual researchers. This is an important aspect of all research. JCAP spans several efforts from fundamental discoveries of materials, material development, but also looking at the fabrication, again, of a solar fuels generator module that will cover kilometer scale areas. So efforts on these scales are performed in parallel with large groups of teams with expertise in diverse areas. So JCAP has organized itself into essentially 8 projects. These involve the design of light capture and conversion materials, the development of new heterogeneous and molecular catalysts, the development of high-through experiments as we begin to create either new light absorbers or new catalysts and high-throughput methods. Not only how do you build these, but how do you test these systems? This directly relates to benchmarking efforts. How do you compare catalysts on equal footing or light absorbers on equal footing? And how do you put all these parts together, molecular nanoscale interface? And once these parts are put together, how do you handle things like proton transport, ion conductivity? This is an area where we have a group that deals directly with membranes and mesoscale assembly, all the way up to prototyping and scale-up. How do you actually think about building a package device that will perform some of the chemistries I've alluded to? So the area of project that I'm involved with is interfaces, looking at how we can interface some of these various components, whether they be light absorbers and catalysts, or components such as the membrane with the light absorber. The key challenge of this group is that many of the things I'm supposed to interface do not exist yet. These materials that are a perfect light absorber, that are robust. But I knew my job wasn't to wait until these things were invented. So what we wanted to do was identify a number of materials that at least have some initial promise. In other words, they have the right optical properties, or they seem like pretty fast catalysts, and begin to think about developing technologies for how to incorporate these at a molecular level to make connections between light absorbers and catalysts. So this illustrates in this slide just some promising materials now from the photocathode side. In other words, not the water oxidation side, but thinking about reducing a substrate to form a fuel at a counter electrode. The optical properties of silicon are quite nice. These are almost perfectly parked in that area where I showed you you'd want in a tandem cell for your light absorption to take place. Other materials we've been looking at include things like gallium phosphide and indium phosphide. So just as a solar flux tells you where you want to park your system in terms of absorbing light, it also gives information on the requirements for catalysis, how fast your catalyst has to be in order to keep up with the solar flux. Again, if you were to say, I want all the photons, well, that's 4,300 electrons per nanometer per second. If you were to capture out to 1,000 nanometers, then you're down to about 2,600. That would correlate, if you converted all of those photons to electrons, to a current just over 40 milliamps per centimeter square in that case. And if you had a catalyst coverage, such you had one catalyst per square nanometer, that would require the catalyst to have a turnover frequency of about 325 per second. So if you were talking about oxygen production in a tandem cell in that case, you'd have to produce 325 oxygen molecules every second. Can catalysts do that? Can molecular catalysts do that? Maybe. You can also lower the requirements by structuring the material. In other words, making nanostructured materials with high surface areas so that you get much more material packed in a given geometric area. So if you can make a material that's 100 times more rougher or 100 times more microstructured, that will essentially lower the demand on the catalyst 100-fold. So now you can talk about having catalysts that operate with a turnover frequency of 3 per second, for example. So just as nature offers a number of examples for systems in thinking about photosystem 2, for example, for water oxidation, it also offers inspiration and design features for a number of reactions that are of technological importance. Things like hydrogen reduction by hydrogenase, CO2 reduction by COD-H and FDH1, as well as complex 1, which handles the NAD plus to NADH. All of these are examples coming out of beautiful work by Fraser Armstrong and Judy Hurst, where they look at these enzymes at an electrode surface and really quantify what's a hitherto unexplored area of how elegant these enzymes are at operating near the thermodynamic potential. In other words, the current densities cut very sharply through the equilibrium potential of the associated redox reaction that these are catalyzing. The downside about enzymes, the downside in regards for human interest and incorporation into technological devices, is that many of them tend to be fragile and many of them tend to be quite large. In other words, they would cover a large geometric area on a surface. That's the reason for the relatively low current densities in all of these. Just as an example, right, I pointed out earlier, can you think about having catalysts with turnover frequencies of 325 per second for keeping up with the solar flux? Well, here's hydrogenase. It has a turnover frequency of 9,000 per second, exquisitely fast. Extrapolation has suggested if you had a mole of hydrogenase, you could fill a Zeppelin in about 10 minutes with a turnover frequency like this. So nature has really designed fast catalysts. The problem is, again, they're big. So a mole of hydrogenase is 200 pounds. So this raises the question, can you look at the key elements, the design features in biological systems, second coordination sphere interactions, how the complexes handle substrate delivery, substrate removal, how they make environments which favor the catalytic transformations that you're trying to drive. The thing about human-engineered systems or heterogeneous systems is there's particularly an emphasis on surface structure. But in biological systems, catalyst involves very, very specific three-dimensional sites. With molecules, you can really form pockets that are necessary for catalyzing the geometric transformations in the excited state. As an illustration of this work, I point to some recent work coming out of Dan Du Bois' group at PNNL, where they've done exactly this. They've looked at building enzymes which capture some of the key elements of hydrogenase nickel active site, in particular a soft ligand environment for stabilizing low valent states of nickel, and these putative amine bases which deliver proton substrate to the nickel. These systems, you can modify the ligand set to make them exquisitely fast. This is an example of a catalyst reported, again from the same group in 2011, that has a turnover frequency estimated at 100,000 per second. This surpasses biological enzymes in terms of their speed, although it's at a relatively high potential that you're driving this. This is an electrochemical process, not a photochemical process, and the overpotential that's applied in many of these examples is upwards of 500 millivolts. Nonetheless, it captures the point. One of the things that I mentioned we've been trying to do in JCAP now is think about the benefit of incorporating molecular catalysts, especially for things like handling CO2 reduction, where the selectivity of your substrate and product is probably going to be really important and may require three-dimensional architectures, i.e. molecular catalysts. So we began to develop a technology for functionalizing semiconductor materials that relies off a UV-initiated process where we can place masked amines onto the surfaces of things like silicon, indium phosphide, gallium phosphide, all these materials I mentioned earlier that have promising optical properties, that then give us these chemical handles to which we can attach molecular catalysts. This is work that's being done in conjunction very closely with Alex Kroshwitz, who's a postdoc at JCAP, and Ian Sharp, who's a staff scientist at JCAP. We've also not only been able to adapt these chemistries to a range of promising semiconductor materials, but have now begun to carry out chemistries where we covalently attach these to very promising and fast catalysts, such as the nickel P2N2 system that I alluded to earlier. These are examples of how we characterize these systems structurally. This is a surface-sensitive IR technique where we can see many of the vibrational modes that's associated with the ligand environment on the surface of either silicon or gallium phosphide, where we've carried out this type of coupling chemistry. These confirm the presence of the ligand, but raise questions of whether the nickel's still present. So there are other types of experiments that we can do for addressing the presence of the nickel. XPS is a nice, again, surface-sensitive technique. And again, in these samples, both on silicon and gallium phosphide, we have pretty strong evidence that not only is the nickel present in a molecular environment, but is also in a 2+ oxidation state. So I think in regards to time, I should get ready to wrap the talk up here pretty quick. This is a slide that I borrowed, or stole maybe you could say, from Tom Moore, when I asked him the question of where are we in terms of progress. And by analogy with solid-state physics, Tom would say we're somewhere between here and here. Well, physics did this in 60 years, and the question is, can we do this? Can we take a leap from here to here? So thank you for your time, and I need to acknowledge many of the people who were involved in some of the research efforts that I shared with you today, spanning many institutions from Arizona State University and Yale University, Argonne National Lab, and the CEA at Clay, as well as some more recent efforts at JCAP. and also acknowledge our funders. And again, I shared with you only a small sliver of the research taking place in artificial photosynthesis, and those who are interested in this area, I can point you in the direction of some recent review articles which touch more so on some of the concepts that I tried to address today with you. Thank you for your time. Mm-hmm. Yeah, interesting question. I can speculate on this, of course, but it's sometimes hard to get at the experiment which pins this one down. For these systems that you're talking about, we want one that handles, I imagine you're talking about the co-deposited systems where we looked at putting a molecular dye down and a redox active catalyst. In that case, we do want one species that handles light absorption and another species that lowers the energy for the photochemical process requirements that we're trying to drive. The thing about that, when you think about it, is that photon absorption of photon by especially a molecular species is a one photon, one complex event. So for every one photon, you make one charge-separated state, whereas many of the catalytic processes are multi-electron and often multi-proton processes. So having these coupled in molecular systems with a discrete light-absorbing species and a discrete catalytic species does mean that these systems, there's an inherent mismatch in the stoichiometry, right, of photons coming in, and that you have to absorb 4 photons within the lifetime of charge-separated state. And that's a difficult thing to think about doing for molecular species. This approach of separating them is one that may be promising. We took this approach in the example that I showed. Interestingly, some of our early studies that weren't reported, we find that the best ratio you can have in that particular system is one where you have 4 equivalents of catalyst for every 1 equivalent of light absorber. Now, that's just the opposite of what I told you might expect if your demand is to have 4 photons coming in to drive something like water oxidation. Interestingly, another system that sort of uses strategy is the one I showed you by Tom Maluk and coworkers, where they've used an iridium oxide nanoparticle that's capped with one of these high potential mediators and a light-absorbing species. And in that case, again, they found the same stoichiometry requirement, a 4-to-1 ratio of catalyst to dye. So I think it's an open question. There are definitely benefits of having things covalently attached and that the electron transfer process between 2 molecules can be very fast. But there's also advantages to having things separated and thinking about controlling the stoichiometry of catalyst to dye. So an open area of research, I think. Thank you.